So, hello. Today we'll be presenting a research project where we investigated how different concentrations of caffeine affect the growth of Saccharomyces cerevisiae, which is the yeast that is used in many lab experiments. This was our first time designing our own project, so I'll go over our background, our hypothesis, our setup, results, limitations, and what all we have learned. A little bit about S. cerevisiae now. It is a commonly used model organism. It's a commonly used one because it grows quickly, it is safe to handle, and it has many conserved cellular pathways. This makes it ideal for studying how cells react to stress. Caffeine in yeast acts as a chemical stressor. So, based on our research, it interferes with several major pathways. The HOG pathway, which normally responds to high osmolarity and stress, the oxidative stress response, which protects cells from reactive oxygen species, the RAS CAMP pathway, which regulates cell growth, nutrient sensing, and division. When caffeine disrupts these systems, it puts yeast under stress and slows down its ability to divide and grow. That's why caffeine is a good molecule for studying stress related growth inhibition. Our research project is pretty straightforward. Does increasing caffeine concentration reduce yeast growth? So based upon our different resources, it shows that caffeine activates stress pathways and it interferes with the growth regulating signals. So our hypothesis is that if caffeine concentration increases, then the growth of S. cerevisiae will decrease. To test our hypothesis, we created four caffeine treatments. We got 0 millimolar, 5 millimolar, 10 millimolar, and 15 millimolar. We began the experiment with a yeast stock culture that was at OD 2.021, and we diluted it to a uniform starting absorbance of about 0.8 for all of our treatments. Even though we aimed for 0.5 originally, our actual value turned out to be slightly higher, and we decided to keep it consistent across all our batches. We measured growth using OD 600 every 30 minutes over a total of 150 minutes, and we ended up with 3 replicates per concentration because that was enough to show us a trend. Our methods now. So our method involved preparing 20 ml batches for each caffeine concentration for 5, 10, and 15. For each batch, we added 4.786 ml of our yeast stock that we calculated, and we filled the rest with growth media. We then dissolved the appropriate amount of caffeine into each treatment. After mixing thoroughly, we pipetted 6 ml into each test tube to create our replicates. And then our tubes were then placed in the incubator at 30 degrees Celsius, which is the optimum temperature for yeast growth. We used a spectrophotometer to measure OD 600. So higher the OD, more absorbance, so more cells in it. We did miss our initial calibration step until the 60-minute time point. So early OD readings in the 15 millimolar treatment were kind of high, but once we calibrated it properly, we could see the readings stabilize a little bit. Like any real experiment, ours had limitations as well. The biggest issue was that the spectrophotometer wasn't calibrated until the 60-minute reading due to lab being super overcrowded. There were many test tubes and there was lots of equipment scattered, which made it easy to mix things up. And we also had a higher than intended starting OD. But even with these limitations, all of this, the core trend in our data was still visible, which made our results valid enough for interpretation. This is our final averaged OD 600 values. There's the values, and then at different time points, I averaged it out for each concentration. So what's immediately noticeable in this table is that the control group, which had no caffeine, consistently had the highest absorbance values overall. The 5 millimolar and 10 millimolar treatment stayed much lower across the whole experiment, and the 15 millimolar treatment did start pretty high, but Abhiraj will kind of explain what happened and what that means. But overall, the raw numbers suggest that caffeine does slow down yeast growth, but the trend becomes a lot clearer when we look at the graph next.
Well, thank you so much, Keshav, for the beautiful presentation. So, as you saw in the previous slide, our raw data table, and this is the growth curve we got from that table. This graph shows the full growth of our yeast over 150 minutes at 4 different caffeine concentrations, starting with the 0 millimoles control, which is the pink line. Now, this line represents normal yeast growth and it is without any stress. We see a slight increase up to the 16-minute mark, which is expected as yeast begins the exponential growth. After that, OD gradually decreases, which likely reflects some settling of the yeast cell or the beginning of the nutrient limitation. But overall, the control curve is highest and most stable, which tells us that yeast were healthy and behaving normally. Then, looking at the 500 millimoles and the 10 millimoles, the blue and the purple lines, these treatments clearly show reduced growth compared to the control. Their OD values stays much lower throughout the experiment. At 5 millimoles, the yeast still grows a little, but much more slowly. At 10 millimoles, the growth is even more suppressed. And the 15 millimole treatment, which is the green line, it is the most interesting line because the first 2 points, which is 10 and the 30, are artificially high. But that's because spectrophotometer wasn't calibrated until the 60-minute time point. So these early values don't represent real yeast growth. But once calibration was done properly about like at 60 minutes, the absorption drops sharply and stays lower than the control for the rest of the experiment. So even though the early values are inaccurate, the trend still shows strong growth inhibition at highest caffeine concentrations. Now, our discussions actually match the results and the literature that we studied. It is known that caffeine triggers stress pathways like HOG and oxidative stress signaling and TOR inhibition. These pathways slow down cell division by shifting the cell focus from growth to stress survival. This is why the 5 millimoles and the 10 millimolar lines stay low throughout the experiment, even though 15 millimoles were inaccurate early on, but once they were corrected, they actually showed inhibition. Now, this tells that our biological outcome was reliable despite our technical issues. Our experiment had a lot of several strengths also. When we calibrated the spectrophotometer correctly, our OD readings showed a consistent trend. We also maintained the same temperature and the media across all the treatments. Something I think could improve the project for future students is better time management between all the lab sections. Now, this is due to the lab schedule not followed by the L1 lab, and due to which L2 lab, which was my section, walked into a really crowded and cluttered lab. These were there were identical tubes everywhere and leftover equipment. Honestly, a 10-minute cleanup time between the sections could have made a huge difference. It would have avoided all the mix up and also prevented things like forgetting to calibrate spectrophotometer. Conclusion. Now, to conclude our whole experiment, our data actually supported our hypothesis. It also showed that increasing caffeine concentration actually reduced yeast growth. Now, this experiment helped us to understand how stress pathways in yeast and actually gave us a very good observations about our lab experiences and how to deal with all the lab challenges that are faced. In the next slide, I have actually written down all the references. For example, the signaling pathways that we talked about, HOG, the oxidative signaling pathway, and the TOR inhibition are all mentioned in these research papers. So go ahead and read each of them. Thank you so much. Have a good day, guys.